I think the thing that's amazing about science and about biology for me is that there's always more to be discovered. And every time we feel like we've figured something out, it seems to open up several more questions. Scientists have appreciated for a long time that once we understood the DNA sequence in cells, if we had a tool that would allow easy manipulation of that sequence, that would be a very powerful kind of technology. Crispr is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way the CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell. It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future.